Okay, sorry about that. I had to start over, record another video. Not start over, but start another video. I had someone come to my door and offer tree removal service. Excuse me, it was OG&E. They didn't offer. They just said they're going to come remove a tree in my backyard. So apologize for that interruption. But here we go. So we're back to rods and cones. We were talking about this is the important stuff that I really wanted to focus on. Those other cells are obviously really important in the process, but rods and cones, for the sake of this course, this is where the money's at. Rods and cones serve different functions. You find them on the retina, and they provide very different functions, but very important functions. You'll find that cones are the cells along the eye or along the retina that allow us to see in color vision, whereas rods allow us to see in vision in black and white, see shadows, shading, things like that. I mean, humans aren't very good at seeing in the dark and seeing it, in dark conditions, but we are, we're not totally blind. So, that's thanks to our rods. The fact that you can kind of walk around your house at night and maybe kind of see some shadows of things enough that you don't just bump and blunder into things are really important. It shows that we just have more cones than rods. It shows that we are a species accustomed to seeing in color, but of course we are accustomed to seeing in darkness, to some degree of darkness. We can thrive a little bit in those conditions. Not like other animals. We would sort of see a different balance maybe in cats. If you have a cat, you know that they are very much like nocturnal animals. They see well at night. You would probably see more of a different balance in terms of rods and cones in this species compared to human beings. So if you have your textbook handy, I want you to try a really interesting activity. It's called the blind spot, and we can actually test. If you remember a couple slides ago, we said there is a spot along the eye, the blind spot, where there are no rods and cones. It's where the optic nerve is sort of leaving the eye, and you can try this activity called the blind spot, sometimes referred to as the disappearing act. So what you want to do is close your left eye and take your textbook, hold your textbook close to your face. Now you should be on page 70, of course, which will have the same picture as here in 3.3. And hold your textbook close to your face, and look at the boy with your right eye. Slowly move the book away until the pie disappears. The pie disappears because it's being projected onto the blind spot of your retina. At that point, the axons of the ganglion neurons collect to form the optic nerve. Note that when the pie disappears, your brain fills in the missing checkerboard pattern, which is one reason that you're not usually aware that you even have a blind spot to begin with. So yeah, your brain will sort of fill in if you're doing this right. It's okay if it takes a couple times maybe didn't get it right. You make sure your eye is positioned in the right way. But what your mind should sort of do is this pie should disappear and the checkerboard is what should replace it. Really fascinating stuff, just kind of showing you the work of all this stuff put together that we just talked about. So how good you are, how good your vision is dependent upon things like genetics, but also is literally dependent upon the shape of your eye. People who are nearsighted, for example, versus farsighted people, their eyes are different. They are shaped differently, physically shaped differently, based on those conditions. And there is an interesting condition that some people begin to actually start to develop, kind of in their 30s, sometimes their 40s, depending on genetics, depending on conditions like that, where the lens of their eyes actually start to grow brittle. They're not cracking or breaking or anything, but they're not as strong as they once were, and it's harder for the lens to kind of focus and accommodate. That's one thing that the lens is very adaptable, allows us to see, but this condition of presbyopia makes it much more difficult to see things that are nearby. Makes it really difficult for your eyes to adjust. Making, again, just part of the, we'll talk about aging and talk about impermanence in later chapters, but, our vision, it's on this roller coaster trajectory. It's great for a while, and, maybe it's great until your 20s, maybe 25 or so, and after that, it starts to go downhill. You need glasses, you need eye surgery, corrective lens surgery or whatever. It's really interesting. Our eyes are really good also at adapting. Think about, I think I've used this example before, but if you've ever walked into a movie theater, it's really dark and maybe at first it's really hard to see. As you're trying to find a seat in a dark theater, maybe you're stepping on people and you accidentally kick over your popcorn or something like that. But then, maybe by midway through the movie or, when the movie's over, you're able to walk out just fine. You can navigate the movie theater just fine and there's no issue. It's just this idea of light adaptation, specifically dark adaptation, where you're adjusting to low lighting. Some more, we're going to talk about color and dimensions of color, before when we're talking about vision. Color is important. We kind of touched on it a little bit in the previous slide, but going back to the whole wavelength thing, the actual color is determined by the wavelength itself, the length between waves itself. And some other properties concern the value, how bright or how dark it is. So, saturation, how intense the color actually is perceived. And some colors have psychological associations with cultural settings. Think about in this country, the United States, you say red and blue together, a lot of people will draw the connotations of politics, of Democrat and Republican. Or, if you just say red and blue, some people immediately want to plug in white, red, white, and blue, the patriotic color, America. So, but if you were to say red, maybe in a different cultural context in like China, maybe they think of, communism or something, some other association of time and place is totally different. Same thing, in Russia or wherever. Here's our color wheel. You've probably seen one of these. You've seen a lot of these, maybe as early as like kindergarten. You have your cooler colors on one side and warmer colors on the other, and this is the spectrum of colors that human beings can perceive. Of course, assuming you aren't colorblind, which we will talk about in a few more slides. I think actually in the next video, probably not this one though. Again, sort of already addressed this. You have the cool colors, the green blue side. It's cooler in temperature visually, and you have the yellow orange side, which is considered warm. Visually it looks warmer. You have pairs that are complementary: red green, blue yellow. We will talk about that again. Complementary pairs. And all of this light is a source of all colors, all the colors that we can see. The light that we can see. So again, back to complementary colors, red and green. Red and green would be considered complementary. I think it said yellow and blue as well. And it's sort of like opposites, kind of. I don't know if it's the direct opposite necessarily. I wouldn't go, it's not that exact, but that's sort of what it kind of appears visually. So here's the difference between mixing light and mixing pigment. Now notice when you mix light, it gets brighter and brighter. Notice when you mix pigment, it gets darker and darker. That is because one is an additive process. You're adding more light. It's just going to get brighter and brighter. When you add pigment together, it actually is a negative process. It takes away. It's a subtractive process, meaning we're subtracting light by doing this. Okay, we are wrapping up. We're getting close to wrapping up part 1 of the vision portion of this chapter. Sorry that I'm having to put it in 2 videos. I did not expect OG&E to show up and anyways, ring my doorbell. So anyways, let's do a quick comprehension check. To read this question, you are looking at it, obviously. After the light passes into your eye, the incoming light waves are recorded by receptor cells located in the A, the retina, B, the cornea, C, blind spot, or D, the optic chiasm. Go ahead and pause the video, check your comprehension, and unpause when you think you have the answer. Okay, welcome back. You should have gotten the retina. The retina is where the receptor cells are located, where we have the rods and cones and all those important things that allow us to see color vision and also black and white. So you should have answered retina. Okay, a couple more questions before we wrap up this video. Truth or fiction? When we mix blue light and yellow light, we obtain green light. That is actually fiction. We actually just get brighter light. The more light we add, the closer we get to a pure sort of white light color. It doesn't actually look like green light. If you add blue paint and yellow paint together, you get something closer to green paint, but not the same with light. It's not the same thing. And let's see, truth or fiction? People can be wide-eyed with fear. I forgot to pause the last one. I apologize. Go ahead and pause now and check your comprehension. What do you think? Okay, welcome back. The answer is, in fact, true. This is fight or flight. You literally, when you are afraid, when you are in fight or flight and panic mode, your eyes get bigger and bigger, wide-eyed with fear, and it can physically, you can see it in your eyes. So we are going to stop here. When we come back, we'll look at after images. We'll do another cool activity that I like to do in person in class. You'll get to do it on your end with your textbook, but we'll pick back up on vision on the next video. So thanks for your time.